morning. Hello, good morning. Good morning, sir. Good morning, sir.
Good morning, sir.
Professor Kak, good morning, Professor Kak. Professor Kak is there? Hello?
Yes, sir.
No,
Join.
sir.
Professor Kak, is he there?
Yeah, he is not joined till now.
He will join. He will join. Don't worry. He is going to join. Sorry, do you have his slides with you? Hello?
Yes, sir. I have the size.
Okay, in the case of any necessity.
Yes, sir.
But I think he will manage. No problem.
Yes, sir.
Okay. He has yet to come, isn't it?
Yes, sir. I am just calling him.
Normally, what is the number of attendees in these courses? Yesterday?
Around, sir, 35 to 40.
Yes.
It varies
That
from
we can
35 to
see
40.
here also. 35 to 40. Mostly they are faculty members, isn't it?
Yes,
In
sir.
different
Yes, sir.
colleges.
In different colleges, sir.
Yeah. Mostly from West Bengal.
No, from South also there are so many
From South,
participants.
yeah.
From South India also there are so many participants.
Okay. And Pankaj will deliver a talk at 3:30? At what
Yes,
time?
at 3:45 p.m., sir.
3:45?
Yes, sir.
Okay, maybe I'll be absent in between. I have some other work.
Okay,
And
sir.
then I'll try to join Pankaj. But I may not be able to attend other programs. Some other job to do. Yes. So yesterday program was okay?
Yes, sir. Yes, sir. So you are present also, sir.
Yes. Reddy was SUM Reddy,
Yes, sir.
DRDO. Reddy was our student. SUM Reddy. Simadri.
Hello, sir. Good morning. Will you join now, sir? Hello. Okay, sir. Hello, sir. Hello. Hello, sir. Professor Kaku will join within few minutes. Hello. Hello, sir. Professor Basu, sir.
কিছু বলছো শরীফ?
হ্যাঁ প্রফেসর কাক জয়েন করছেন।
আচ্ছা।
আমি কল করেছিলাম, জয়েন করছেন।
Good morning. Good morning from Indian
Good morning, sir. Good morning, sir.
standpoint. Indian standpoint. Yes, good morning here. Yes.
Sir, please unmute your mic.
Sir, please unmute your mic, sir.
Yeah. Good morning.
Good morning,
Yes.
sir.
to everybody.
Good
Good morning, sir.
We'll wait for some time
Oh, sure.
for others to join.
In the meanwhile, can I share my screen?
Yes, sir. Sure, sir.
You can share this.
Is it visible?
No, sir.
No, it is not visible.
Okay.
It is not there.
Is it visible?
Yeah, it is going. Yes, yes. Yes, yes. It is there.
Okay.
Hello?
Yes. Should I start?
No, you cannot start
Yes, sir. Now it is visible, sir.
because your
Sir,
first
should we
slide.
start?
No, I think first slide you can start, but before that I have a comment to make.
Sure, sir.
Your first slide is not correct. Please read S.K. Kak, then former professor of
Electronics and Communication Engineering.
no,
IIT BHU.
it is electronics engineering.
Yes, sir.
Because I'm not a researcher. I'm a just I find this sort of mistakes. Number two.
No, because
No. It is accepted. It is electronics engineering department, not communication. Number
Thank
two,
you.
because
Thank you.
I cannot follow your lecture. Your subject is not is beyond my comprehension.
No,
But,
not
but
at
one
all.
thing,
It is
again,
it's
there is a
totally
mistake. There is a mistake.
what?
That mistake is, as I'm going to introduce, Professor Kak is not only professor, he was vice chancellor of 1, 2, 3, 4. Let me name.
Five.
First is vice chancellor of UP Technical University. It is known as something. What is that name named as? Professor Kak help me
Mahamaya
to.
Technical University.
Yes, that is the UP Technical University. He was vice chancellor. This is number one vice chancellor.
Yeah.
Then number two vice chancellor of. Now please tell. Either you tell or Sharif Pal will tell. Vice chancellor of which university?
Chaudhary Charan Singh University.
Information technology.
Chaudhary Charan University. This is number two vice chancellor. Number three vice chancellor.
Of Agriculture University in Meerut.
Number four vice chancellor.
JP University of Information Technology, Noida.
I don't want to continue any further. Please now you go ahead with your slides. Okay.
Okay.
So this is my
Thank you.
introduction. Okay.
Thank
Fine.
you.
So first slide
So,
I have correct.
should we start, sir?
Yes. Now you can read his biodata. Deepta,
Okay.
Thank you so
Yes,
much.
sir.
you can read his biodata. Okay.
Yes, sir. Yes, sir.
Yeah.
Good morning, everyone, and welcome you all in the third day of the Faculty Development Program on recent trends and advances in electronics. So, today's session will start with Professor Dr. S.K. Kak. Professor Kak was born in Srinagar, Kashmir, and he has joined as faculty in IIT BHU in December 1971 and became reader in 1978 and Professor of Electronics Engineering in 1985 and has held many other responsibilities as Director Computer Center, Chairman of Telecom BHU, and the President of IT GEMCAN. Since his student days, he was active in the engineering model making activity and learned how theoretical knowledge is applied to solve real world problems. He honed his engineering skills and translated many ideas into active products that have won him many awards, prizes, and two patents. As faculty, he established Communication System Lab in 1973, Optical Communication Lab in 1978, Digital Communication Lab in 1979, and was also instrumental in starting the MTech program 1971 in communication system engineering. He has completed 6 R&D projects for DAE, AICTE, DRDO, MIT, and ISRO as principal investigator. He has contributed variously in electronic product designs, technology reviews, and helped many government and private industries. Professor Kak was director of JP Institution of Information Technology, Noida from 2003 till February 2006. He has been an independent director on the BSNL board from February 2008 till February 2011. He was appointed as the Vice Chancellor of Chaudhuri Charan Singh University, Meerut, 2008, and then Vice Chancellor of Mahamaya Technical University, Noida in 2010, where from retired in November 2013. He then joined the JP University of Information Technology, Solan as Vice Chancellor in March 2014, which he left in May 2015 and is now working as a freelancer since then for promoting skills development using innovative techniques for training and certification. He has been member of selection committees of various national organizations, expert and co-chairman of RAC for selection of scientists for DRDO, ISRO, and DAE, including AICT, executive committee member and UGC expert member and member of NBA Governing Council and chairman of NWRO of AICT. At present, he is the chairman of advisory committee on Prakash, the student learning assessment platform for the E&T management and MCA students of institutions and colleges affiliated to the AICTE. Professor Kak today will give a lecture on bio-nano interfacing to bioelectronic medicines. Welcome you, sir.
Thank you, Deepti. Thank you so much. This is all the baggage that one collects over the period of time. I am so grateful to Bidhan Chandra Roy College, to its members, and to all those who are present. I wish them a very good morning and hope that we will share together a journey of a few things that India needs to do in the bio-nano interfacing and bioelectronic medicines. My major acquaintance with, I'm not basically a biological scientist, I'm basically electronics and communication person, and have been throughout my life involved in electronics, instrumentation, communication, and systems. During the COVID period, I had a very strange thing in September 2018. I attended a seminar in US wherein very famous scientists were present, and they started talking in terms of that no one in future will die of old age. This was the title of the seminar, and Ray Kurzweil was the convener. And the major aim was that there will be so much of ability for people to do genetic engineering, to do interventions in through biomedicine, interventions through other particular processes, that the normal human being can be sustained over a long period of time through stem cells and other particular this thing. And lo and behold, in January 2019, everyone knows what happened. SARS-CoV started, and that created a havoc in whole of the world. We have more than 5 million people dead all over the world. Almost each one of us has gone through a tragedy. We have lost someone, some friend, some relative, someone. And that put me, during that particular period, I got interested in understanding what is this SARS virus, how does it affect, and things and so on. So that's how it started. And for this particular talk, I took this bio-nano interfacing to bioelectronic medicine because I believe India has a very strong opportunity to do this particular research, to present and develop new products, new services, new technologies, and do basic science research also in this particular area. There have been 4 major deficiencies because of which we have not been able to go into bioelectronics as such. Number 1 deficiency has been that the major source for development of bioelectronics was the enormous amount of development that happened in electronic circuits, devices, and integrated circuits. It has gone level to almost 5 nanometer now, that is in 2020. And at the present moment, labs are working or fabs are working to go to 3 nanometers, and even 2 nanometers is being tried. So you can understand that Moore's law in certain particular cases has been exceeded, and in even these particular expectations are going to go ahead. However, there is a limit to which it will go, but that has not arrived as yet. So having billions of transistors on a small chip, and in the meanwhile, we in India do not have a fab. We have a human resource that is capable of designing ICs, designing circuits, designing systems, doing hardware design, doing software design, doing other particular this thing. Many of our people globally are working on these particular things, but unfortunately in India, we do not have a fab. We had some smattering of it in Chandigarh, but that also is now defunct. There has been a certain this thing process that has been developed in by DRDO in Bangalore, but that's also not in the area of silicon. That's basically germanium and gallium arsenide. Now, the second thing that simultaneously happened was that there was an enormous amount of growth in gene mapping, gene understanding, and many of the researchers went down to the point where we are now able to understand what happens in a cell, how cell interacts with the world, and how it can be manipulated, how it can be changed, or how it can be even redirected to operate different functions. So intervening at the genetic level, at the protein level, at the genomic level, that is where we are today in terms of biology. So these two things are coming together, and that is what makes bioelectronics a totally different area today. And that's what interested me, and so I'm going to talk a bit about that. again, I would say that I'm not a biological scientist. I'm basically electronic scientist. So my focus would be more towards electronics rather than towards biology. So these developments have basically started talking in terms of, can we deliver healthcare? And that healthcare has to be not only in terms of being able to diagnose and being able to have that diagnosis on a massive scale, being able to create medicine that is targeted to the individual's gene profile, is being targeted to be his particular organ dependent design or organ dependent failure or organ dependent deficiency, or maybe some deficiency which is generally in the body, let's say diabetes or high blood pressure or something like that. So generating healthcare, generating medicines for that. Then this drug design is targeted drug design, and how we can gradually not use drugs for flooding the body, but can we target those drugs, target the drugs to certain particular location where the problem is and where the disease is. So that targeted drug location and targeted drug delivery is another particular area. Besides healthcare and medicine, it's also important to understand that we can have enormous amount of understanding about forensics, national security, environment, food and agriculture, and even other opportunities in manufacturing technology. We are understanding so much from biology about how aggregation can be made, how things are constructed, how we can use bacteria, how we can use insects, how we can use insect-driven knowledge in order to design other mechanical things or other robotic things and other particular things. We are today designing certain capsule-based micro and nano robotics that can travel in the body and try to understand what's going on in the body, or alter certain particular situations in the body. So nano and micro 3D assembly and interfacing materials, etc., is going to be a very, very important thing. But at the same time, there is another particular area that has come up very strongly, and that is surface chemistry. The moment you try to put something in the body, its mechanism immediately understands that it's a foreign body, attacks it, and tries to throw it out. So how to neutralize that? How do you make a system that is non-invasive, that is camouflaged, and that cannot be detected by the human body, or if it is detected, it will not be thrown out? So it is that particular kind of in vivo ability to design these particular things, making them stretchable, flexible, making them biocompatible, making them non-invasive, and most important of all, where from do they get the energy to function? So building in energy resources onto the particular device. Similarly, there have been things on lab on chips, DNA chips, gene chips. So for genomic, proteomic, and DNA profiling and system biology applications. I have talked about this, but the basic understanding is that tomorrow, the future of life, is it in our hands? Can it totally be controlled? As I said, the beginning of the particular talk, that is it possible that there is no death? Is it possible that we can stop biological degeneration or degradation? Those are particular esoteric questions. There are so many ethical and other particular aspects to that. But we are trying to talk in terms of bioelectronics at the same time, trying to do various particular areas where we try to understand molecular cell electronic interfaces, understand cellular responses and their variables to simulations like electrical, mechanical, optical, chemical, thermal, and the like, ability to collect and analyze essential data on the state of biomolecules and cells, how they are, their electrophysiological parameters, their biochemical parameters, how they are functioning, what is going on, and things like that. Can we monitor that? And at the same time, can we understand, because when a gene expresses itself, it creates certain proteins. And this proteomics, the amount of protein is in femto level or sometimes alto level. Can we have probes which will be able to measure that quantity, that here the gene has expressed and this protein has evolved? So is this possible? So this is the range in which we are trying to work for the future. Basically being able to generate biological sensors in situ, in vivo, in vitro, in the environment, in other particular environments, on animals, on human beings, and other particular areas, magnetic field sensors, electronic sensors, electrochemical sensors, neurophysiology, engineering microstructure, neural network structures, and of course, learning from them to design other particular things. So the combination of nanosystems, the combination of cellular systems, the combination of being able to join them together, make them work together, is the area where we're trying to talk in terms of the future of bioelectronics tree. And simultaneously, what's happened is that a lot of investment has gone into AI and artificial intelligence, machine learning, immersive reality, and things like that. So we can train today doctors, engineers, or scientists, or other particular people in novel areas through immersive reality, and we can force machines to learn, and that learning is coming through artificial intelligence. Now, I'm not trying to say that artificial intelligence at the present moment can replace human intelligence, but there is now a novel step that has come up, what's known as artificial general intelligence. Till now, we have been developing algorithms, we have been developing software, we have been developing tools and techniques for a particular application. For one particular zone, we have this artificial intelligence tools. But can we generalize them? Can we start talking in terms of a general artificial intelligence ability where the system can learn as a child does, where it has inquisitiveness, where it has the ability to imagine, or where it has the ability to understand its environment in which it is functioning, interact with it, try to learn on the job, and things like that. So this particular artificial general intelligence, how is it getting involved with this particular bioelectronics, is at 3 levels. Number one is where we are trying to talk in terms of, okay, let's see that if we are taking all this particular data of an individual, on the basis of that, do we have algorithms to know that from his blood chemistry, from the expression and the genetic map, from his genomic studies, do we have the capability to understand that if there is any internal threat to his body, is there any cancerous cell moving around? Is there any blood glucose that is toxic moving around? Are there any molecules or other particular toxic substances that are going on? So that particular kind of thing, are we able to learn? And therefore, what we are trying to talk in terms of is, are we at the present moment having the capability to have a separate brain, human-like brain, which can understand these particular things, which can reprogram themselves, which can train themselves for this particular application. So therefore, the third aspect that has come into the biosensors, biotechnology, biosystems, and bioelectronics is the introduction of artificial general intelligence. The research priorities in bioelectronics have basically been, the drivers have been, can we create prosthetics? We have known that there is a very large number of people who lose their limbs, or who lose their organs, or who lose their particular skin or anything, tissue, because of certain particular this thing, and can we develop that artificial those prosthetics, whether it is limb, hand, whether it is artificial larynx, artificial retina, artificial other particular this thing, artificial skin, which has become a very, very powerful tool now, and it is already available. And at the same time, can we have disease prevention, including treatment for Alzheimer's and treatment for Parkinson's, treatment for cancer, being able to detect early cancer molecules and then take a control and have released either chemotherapy or other particular therapies in order to prevent. So this disease detection, disease prevention, and other particular things, these are the major drivers which are promoting bioelectronics. Now the devices that are coming up, as I said, lab on chips. What is a lab on chip? I will cite it very brief. Protein and DNA chips, imaging including cellular analysis, cellular mapping of biophysiological systems, of biochemical systems, of biointeractive systems, and other particular things, and then doing this in on remote systems. Measurement analysis of non-invasive physical sensing, concentration of analyte and metabolic systems, real-time and time-dependent measurements, single biomolecule detection. This is one of the greatest challenges at the present moment in terms of single biomolecule detection. And this is not only for the lab on chip, but this is also for in vivo and in vitro applications. We are not able to, at the present moment, to understand when a gene starts expressing itself, and we will not be able to understand that unless we know when it has started expressing and may created a protein or created other genomic experiences. So then technologies are molecular recognition, signal processing algorithms for those detections, DNA sequencing, fabrication, including pattern recognition, pattern understanding, pattern evaluation, synthetic pattern generation to control multiple systems and make that happen in a harmonious, synchronous, and effective way, and thin film technology for the conformal wearable systems. Am I going too fast or is it okay? Hello?
It's okay, sir.
Okay. So what is the application of bioelectronics is basically drivers, as we have said, we want to have early detection of heart disease, cancer, stroke, and we want at the same time to develop electronic systems that can prevent these things happening by disease treatment, respiratory, diabetic, and kidney. Microdialysis systems have now come up since 1999. They are not very, very effective. They are not as effective as dialysis machines outside, but they are working, and there are people who are using those. Similarly, artificial retina, we have artificial larynx, we have cochlear transplants, and we have other particular this thing. We have come up with artificial nose, even certain particular, there's not been much success in taste. So artificial tongue is not a possibility, but there are small artificial lung, artificial heart, and things like that. The greatest problem at the present moment is in liver. We have not been able to replicate any of the functions, major function of the liver. Measurement analysis, rapid analysis, DNA profiling, proteins and cell metabolics, multi-channel sensory inputs. This is one of the major hurdles in terms of analysis, because when we try to understand what is happening in one particular patch that we have put on a semi-permeable system, but is that a definitive understanding of the whole body. That's not possible as of now. So multi-channel sensory systems are an important this thing. In vivo measurement and analysis, DNA proteins and cell metabolites. Devices, we have bio-DSP. Well, now bio-DSP is something which is very, very strongly emerging. We are trying to understand how genetic memory gets transferred, how it does certain amount of cell level processing, and whether that can be done when we are trying to develop artificial retina, or whether we can have direct transplant or implant in the brain for those people who are blind right from birth. So micro energy sources, because as I said, that if all these particular tools are to circulate in, are planted inside. Say, for instance, at the present moment, there's many of the people use our heart triggering and pacemakers, and for control of arrhythmia and control of other particular things. So we do give pacemakers, but these pacemakers are generally having some implants inside, or there is some implant inside which is connected to the outside world through either wireless technology or through Bluetooth technology. In either case, whatever is implanted inside, that needs a power source, and most of the time it is lithium-ion battery at the present moment. But can we have fuel cells that are based on cellular mechanism? Can we have energy systems that are based on the movement of the body? Can we have this thing based on the natural metabolites that are available in the system? So those are the kind of micro energy sources that we need to develop, and nano delivery devices which will be able to have the ability to function as synthetic pancreas or artificial pancreas, synthetic dialysis system or dilate process, and things like that. So, and technologies are wireless networks, 3D integration, the technologies that we can learn from there. How wireless networks can be used and converted. I mean, most of us know that the ability to understand how to make mobile cell towers work in a configuration so that there is a total handshaking all over by multiple people at the same time, came from a certain amount of biological understanding of how biological systems work and how they hand over molecules and other particular metabolites. So anyway, they are known as cells, and the name is not just for the sake of it. It has a real meaning. 3D integration. 3D integration is coming very strongly because we're talking in terms of printing things, printing many of the prosthetics, printing many of the devices that are going to be used for implants, printing many of the things that are otherwise, I mean, 3D printing is a very, very common thing now. And how we can create multiple systems that have biocompatibility at the same time and are natural supplements or natural part of a human body. So two-way interface with biotissue and being able to transfer information, being able to transfer chemicals, being able to transfer molecules and things like that. So cellular and molecular challenges that are there in this particular development are we able to develop systems which can rapidly determine the whole genome and RNA sequence? As you know, that at the present moment, we are not able to do this genomic studies, which it takes at least by the computer, even after feeding the whole data, it takes at least few hours in order to do the genomic analysis or RNA sequencing. So can we do it in situ almost instantaneously? That's one of the challenges. quantify the up and down regulation of multiple genes over many time points? In other words, are we able to understand how genes are being changed because there is a certain intervention, or there is a certain exposure, or there is a certain excision that's happening either through treatment or through cancer or through other particular interventions? Can we do this time point analysis in a particular thing? The exact applications, there are huge exact applications of this, and identify biomarkers for the disease states. DNA markers, RNA markers, proteins, polysaccharides, or metabolites in both serum and actual blood and tissue samples. Now, here, when we are trying to talk in terms of in serum and actual blood, that we would like to avoid, but serum is a very real possibility. But tissue samples is in vitro, it's not in vivo, and therefore that has a lower amount of category as compared to the actual blood and direct biomarker identification. Identify, quantify biomarkers in real time at sensitivity levels equal to or better than existing lab techniques such as enzyme-linked immunoassays, and also be reusable. Now, this is a tall order. This is probably one of the most challenging research area, because moment you have this kind of biomarker identification, as I said, these biomarkers or biomolecules can be at the level of pico or alto concentration, and yet there will be so much of noise, so much of other particular metabolites, or so much of other particular molecules and other things, that it will be very difficult to identify. An example in point is that those which are continuous glucose monitors, the continuous glucose monitor, even if it is implanted or even if it is semi-implanted, has a problem that the tip cannot continuously measure the particular this thing, because there are so much of other particular this thing. I mean, what influences the oxygen level, the other metabolites that it is measuring in order to understand how much glucose level is there in the interstitial body fluid, and at the same time that is impacted by the other particular portion, and that attacks, and therefore the kind of measurement that we have is not proper. So this is this thing. So monitor multiple intercellular events in real time. So once you get into the cell, are we able to understand that in this cell, what all things are, what electrophysiological changes or parameters are changing, what biochemical changes are happening, and are we able to instantaneously and monitor it in real time? Now, moment you start monitoring something, moment you start understanding either the electrical potential or other things, it has its own impact on the very thing that you are trying to monitor. So that is another particular this thing. And it fouls, biofouling is a very, very important problem in any measurement system used in situ, in vivo, or in situ. Simultaneously determine membrane protein structure and function in vivo-like environment, identify the chemical and mechanical cues that drive stem cell differentiation, recapitulate, this is again linked to the genetic expression and the proteomics and genomics, recapitulate physiochemical cellular microenvironments, and implement ion channel-like devices as sensors for improved molecular recognition and sensitivity. So topical area framework of challenges is basically we do not have the biological knowledge, and therefore the key technologies are missing. What are the key technologies? The key technologies is a very wide band in situ source that is biocompatible, that is non-invasive, that has enormous amount of bandwidth, and can at the same time identify and recognize only that molecule. You know, when I started talking, when we started talking about this particular thing, you will remember that a virus can identify the real cell and bind to it. So we do not have any kind of such kind of a device in electronics or in biosensors that can do that kind of replication. We have certain RNA probes, but it is far, far away from that. So biotic and abiotic interfaces, like I said, and ethical, regulatory, and safety features. Moment we have any nanosystem into the body, it has its own problems, it has its own toxic effects, and that we do not know. So rest of the things we have covered, so I'll bypass this slide. Okay, now we come to the real one where I want to focus, and these will be the three most important slides. So we have variable, semi-implantable, and fully implantable bioelectronics systems. So when we try to talk in terms of the variable, what we are trying to talk in terms of what are the patches on the body, what are the things that we can wear. Many of you must be knowing that we have this particular Apple smartwatch or other particular smartwatch that tell you about what's your heart rate, what can also measure your blood pressure, can sometimes even measure how well you are doing, and can retain certain particular function, respiration rate, oxygen levels, and other things. But this is basically derived information from the particular algorithms that have been built into those particular watches. So the variable implantable bioelectronics is basically that has the ability to understand the biological function of the system, but at an algorithmic level, not at the real data level. The real data that it is getting while you are walking, while you are moving, while you are running, and things like that is only a certain particular surface level information. Now, the problem with this is that, number one, this has to have a very tight or very good fit onto either your wrist or your arm or wherever you are applying it. And that has to have a certain amount of connectivity to the skin. Now, this skin connectivity is also, as we know, there are stretchable membranes on the body, but these particular watches or other things do not conform to that. And therefore, the conformability of wearable implants is a very important issue. And that is what is lacking at the present. So, we come to the next level is the fully implantable bioelectronics. Now, when we are trying to put fully implantable bioelectronics, we gave the example of pacemaker, or we gave the example of putting some trachea, cochlear, or artificial retina, and directly implant certain electrodes in the brain in order to translate that. This particular device has come in 2021 and has been tested in certain particular laboratories, but it's not a commercial product as yet. So when we are implanting anything in this thing, the three major problems come: the biocompatibility, in vivo retention of the same this thing standard, because you cannot take it out again and plant it again in order to make standardization, in order to make that it sticks to the specifications and it is working as designed. So this is probably one of the major problems with fully implantable bioelectronics, that its characters should not change. It should have a long life. It should not be able to be corroded. Should not at the same time impact the tissue wherein it is implanted. Normally what happens whenever there is a particular implant, that starts developing a tissue heating, and that tissue heating slowly kills the tissue, and therefore the interface stops. Same thing happens in many of the pacemakers also, and therefore they have to be taken out after some time. And at the same time, their battery does not last for full time, and therefore also what happens is that they have to be taken out and a new surgery done in order to implant another one. So fully implantable ones have these three particular problems. rejection, tissue damage, and energy source, and at the same time being able to provide sustainable, continuous, standardized responses in the in vivo system. Compared to this, now between the two is what is known as semi-implantable bioelectronics. This semi-implantable bioelectronics has both these particular, the in vivo application and cell application, whole matrix is there. And these are basically where we are trying to talk in terms of electrical recording, biochemical sensing, drug delivery, and this particular, when we are having this kind of a system, it is basically implanted, fully implanted. When we have this kind of a system, it is partially implanted or semi-implanted, and therefore this is the major difference. Now, what is the advantage? Is that semi-implantable systems can have energy sources from outside, do not need to have transmission of information through wireless or Bluetooth. Therefore, the impact of heating, etc., on the tissue is very, very minimized. And finally, the most important thing is that what is implanted is a very, very small micro needles, and therefore their rejection possibility is much, much lower. So the first system that we start talking about is a nanowire biosystem, where we have basically three functions. Number one is what is known as a PBS, or photosynthetic biohybrid system. This photosynthetic biohybrid system is basically that normally wherever you are in general, there is some light available, and that particular can be converted into energy, and that energy can be then coupled to the whole line, and at the same time what we sense can be sensed through this nanoscale field effect transistor, and for high resolution physiological signal recording. And at the same time we have these microbial fuel cells that use the organic structures on which it is sitting to generate this thing. So we have three important aspects, either through photonics, or through biofuel cell, or through nano contact FETs. We generate these particular fuel cells, they can generate sources. So this is basically interfacing, where we are trying to talk in terms of biosystem interfacing with a nanowire. Now, these nanowires we're talking about are basically either nanofibers made from silicon, made from carbon, made from gallium arsenide, or made from titanium, or made from gold, silver, or other metals, and then continuously being made into this particular kind of this thing, means that we most of the times we are using silicon, because the processing technology of silicon has matured, is very well established, and we can develop that both from the point of view of a substrate to any this thing, the kind of heights and the kind of dimensions we're talking about. It's absolutely controllable, completely this thing, and we can develop silicon dioxide coating, and other particular coatings on that. So that is why most of the time we'll be talking in terms of silicon nanowire. So here we're trying to talk in terms of this nanowire biointerface, which is trying to see how a bacteria is being impacted by this nanowire, and this nanowire is in touch with this particular bacterium, and the electrical interaction with the bacteria is being reported by this particular nanopillar. And this attachment with multiple molecules of the bacteria or their movement from one point to another point, or their separation from this, everything can be monitored and can be recorded through this nanowire biosystem. And this is basically how we do it in a fluid. So in a fluid, if we have a bacterial colony, then we can have these nanopillars, and how they interact with the particular pillar is this thing. The basic structure, this nano pillar, is of the order of approximately 6 micrometers and 100 micrometer height. And this bacteria itself is approximately of the order of 3 to 5 micrometer, or can be up to 10 micrometer. Now, when we are trying to talk about this kind of a system interfacing, why we are talking at this, there is not much of an understanding in terms of the impact it has on the bacteria, but we are trying to get the data from the bacteria. This is not true when we are trying to talk in terms of tissue that is very, very large. So in that, what we can do is that if there is a particular nanowire, a single nanowire, the total data that it can capture from that particular one molecular point or one cell is limited. So what we can do is we can have these multiple this thing, and that is what is known as nanowire array. And this nanowire array has multiple geometries, and these multiple geometries can themselves act in certain ways in order to improve the interaction with the fluid, improve the interaction and the collection of the information, and improve at the same time how we interact with the environment in which we are acting. Especially here, we are trying to talk in terms of a cancer cell, where the topography mediated cancer cell, how it is impacting and how its sensing is happening with one and other particular ganglion cell. So this particular this particular part has been expanded, and here the neuron contact in brain, or neuron contact in a particular this thing, has been cultured on this thing. Now, at the present moment, in order to measure this particular thing, we can have all these particular things develop on a nanopillar or on a nanowire, and therefore we can see how it is interacting, how that particular thing is happening. Now, when we are trying to talk in terms of this nanowire and piercing a cell in the by this particular thing is probably one of the most difficult things, and that is done through phoresis. And this can be electrophoresis, optical phoresis, mechanical thing, or other particular things. So placing the nanowire inside a cell or putting nanowire into a cell itself is a very high level of technology, and therefore that interfacing becomes one of the most important ideas. So in unlocking the potential of nanodeoxide, the improving cell adhesion. The cell adhesion means that when we are trying to talk in terms of piercing the cell or touching the cell, when we are touching the cell, we are only talking of the extracellular sensing. When we are piercing the cell, then we are trying to talk in terms of intracell interfacing. Now, when we are having an array of these, the array, we cannot change the array dimensions, but what we can do is we know what is the intercellular distance, and therefore the whole design of the array is done in that particular format, and then the culture of the biological material is also done in that particular manner. Now, when you have a cancer cell, this cancer cell is developing on a particular metal pillar, and this metal pillar itself is having the ability to adhere to this particular cell. And if this adhesion is not proper, or if the penetration is not proper, then the kind of signals that we'll get and the kind of biomarkers that we'll be having will not be up to the mark. So in order to improve this, what they have done is they are trying to talk in terms of a kinked nanowire array. This King narrow array is basically trying to talk in terms of, I'll just let me show. Okay. Strict narrower is basically we have either a U type this thing or a V type nanowire structure or a W type nanowire structure, basically defining which kind of a cell we are trying to penetrate and which kind of cell measurement we are trying to make. So when we are making this V-shaped or W-shaped or this thing, these kinked nanowires will be working at an angle which will be defined by the particular pillar, and once this particular thing is known, then this surface modification or additional potential charge that we observe because of the kink, because it continuously surrounds it, then that particular thing becomes much more elaborate. It improves almost 100-fold the potential that we are able to measure and the bio signal that we detect. And since it is engulfing the whole cell or engulfing the structure, therefore it can give better intercellular signals than the simple nanowire probe. The second important thing is that in the intracellular region, the cell member fusion and the design of the particular kink, that itself defines how we can modulate the kind of influences that this particular nanowire is having on the biological cell or biological series of cells. And these series of cells in turn are being understood. Say, for instance, this cell 1 and cell 2, this is the interface between the two, and this interface we are trying to probe through the particular nanowire. And this particular nanowire, you can see this particular probe here, and just a magnified part. And this particular system interface is basically developing between one cell and second cell. So in this particular way, we can have intercellular this thing. If we have an array of these particular this thing, so we can see what's happening in one cell and then what's happening in another cell, and how much time it takes for the process or the stimuli or the molecule or other particular biomarkers to move from cell to cell. So these are the kind of interfaces we try to talk in terms of nanowire testing. Now, before we move ahead to the next slide, I would like to now try to talk in terms of what are the medical applications of these particular this thing, and at what level of technology are we at the present moment. The level of technology, basically, we are able to measure at pico level, because the proteins are at the pico level. The bioanalysis profiling is possible at that particular level, and where we are trying to collect data points of approximately 10 to power 5 or 10 to power 6 per unit of the implant. And this particular means that the bioanalysis profiling will have that much amount of data points. And so in order to take this analysis, and in that we understand at the picomolar level is probably the major problem at the present moment. And when we are trying to talk in terms of gene expression profiling, we're talking of alto level to zeta level, and the data rates are going to be 10 to power 8 to 10 to power 9. So major application areas are in neural implants, artificial pancreas, vision and hearing system, disease prevention, including degeneration and cancer, lab on chip for proteomics and genomics, electronic protein and DNA chip, imaging including cellular, subcellular, this thing, and telemonitoring. non-invasive physical sensing, vital functions inside the body or on the surface of the body or in terms of what is implanted. As I said, we can have capsules which are in the lipid form running through the body, and they are in turn collecting the information and then putting it out. But that technology is the most difficult at the present moment. Single biomolecule detection, including mass, size, chemical, optical, and other properties, molecular recognition, which molecule it is, what are its characteristics, and how it can be analyzed, signal processing algorithm for real-time biomonitoring of cellular functions, DNA sequencing in vivo and in vitro, and nanofabrication of these particular thing, and using thin film technology. So what we are trying to talk in terms of is that now we are coming to something what's known as bio nano system biology and bioelectronics. We are trying to go to a level where the biological system is interacting with a foreign body. Now, when this foreign body is interacting with it influences it, and it at the same time tries to understand how the body is functioning. Now, we have at the present moment, as we said, cytometry devices with high throughput and accuracy. Just a minute. Hello. Hello. हाँ, मैं अभी एक मीटिंग में हूँ, बाद में बात करता हूँ. हाँ, I'll talk to you later, okay? Sorry. So what we are trying to talk in terms of where is the question of system biology coming into bioelectronic medicines. Now, system biology basically tries to talk in terms of that everything that we are covering in a human body or in a this thing is a system. So what happens at one particular point influences the whole system. And therefore, when we are trying to make these measurements or make these interventions or understand how one cell is functioning, it does not mean that we have to keep them in isolation. So this basically defines that we measure at a certain particular point, but it is impacting the whole system. So when we are trying to talk in terms of this kind of implementation, we have three major hurdles. Number one hurdle is that the amount of data that we have to handle, and that can we handle in real time, or are we handling it, collecting the data, analyzing it, and then taking steps on the basis of that, and then giving a certain this thing. Say for instance, let me take a very simple example of a continuous glucose monitor and a insulin pump. Now, when we are trying to talk in terms of a continuous glucose monitor, there are three ways in which it can be done. Number one, we can monitor the blood level glucose, and this blood level glucose, therefore, a particular probe has to touch either a blood vessel or has to take a blood this thing. Nowadays, we know everybody can do his blood glucose, not very reliable, but can do a blood glucose monitoring at home by using a probe which takes a drop of blood on a sensor, and that sensor we can tell you how much amount of glucose is there in the blood. Now, the problem with these is the cause of infection, the number of pricks that you have to have, and which can at the same moment you cannot have it continuous monitoring. You have to have it monitoring only at a certain particular time. You can do it at best twice in a day or something like that. But if you want to have continuous monitoring of this thing, so how does it happen? If you try to put a probe or a nanowire into a blood vessel, then you can probably monitor it. But the data that is coming out, that the probe has to be there. Now, as the body moves, as the probe is this thing, does it remain in contact with the vessel? Does it impact the vessel itself? Will it not stop the clotting? Will it not start the clotting at that particular point? Is the non-invasive system that this nanowire is having impacting the particular tissue around it? And for how long can you use this particular plant for there? And most important of all, the particular coating or the enzymatic action that it is having with the kind of coating that it has, how long can it do the real measurement in a standard manner, and what will be the change in that over a period of time. Now, therefore, this continuous glucose monitoring system, people have started talking in terms of not having to go as deep as to a blood vessel, but doing it in upper dermis level, in the upper level with microneedles, which are not more than 10 to 20 micrometer deep. So when you have this particular patch on your body, and therefore what it measures the interstitial fluid and what is the glucose concentration in the interstitial fluid rather than the blood. So when it is measuring that, obviously there is a time lag between the interstitial fluid and the blood glucose, and this has been found to be from anywhere varying from 5 minutes to 10 minutes. Now, when we are doing this, so we are continuously monitoring this particular interstitial fluid, this thing, and how we are monitoring it? We are monitoring it by having a certain amount of correlation between blood glucose and the metabolites in terms of certain particular oxygen level and hydrochloric acid level. So when this particular acid is there, you are measuring it. Doesn't the other body function molecules also impact that? And therefore, isn't there an error in the measurement? And this error in the measurement is the major source that continuous glucose monitoring has become a very, very different this thing. So in a system biology where the system has multiple sources of influence as compared to a single source, therefore the ability to create filters, films, and masks that are masking other particular fouling agents as compared to what you want to detect, or you have to have a single ability to have direct beaming of that particular molecule only, or that particular reactant only, and then catalyze that and measure the electrical impulse or whatever you are trying to measure in terms of the connectivity of that metabolite. So this particular thing tries to tell us that if we are trying to measure the glucose level continuously, and as per that we are trying to pump in through an insulin pump into the body, so that the body it acts as a synthetic pancreas or an artificial pancreas, then this particular algorithm is what is part of the system biology. This thing. Similarly for microdialysis, similarly for creatine evaluation, kidney functions, this thing, and other particular devices that are being implanted. So at the present moment, there is one more important area which has been understood, is that there are certain particular markers that try to talk in terms of tumor cells. Can we detect those tumor cells? And if we detect those tumor cells at a very, very low level, which are flowing in the blood, but they are not directly available in the interstitial fluid, but is there a possibility that we can detect them even in the interstitial fluid at very low level? So then we can have that particular kind of identification of those tumors and this thing at a very early age. Immunoassays, I mean, basically being able to understand what proteins are there, what particular gene profiling is being done on time, in real time, and Masuli at the same particular point. That's another particular system biology approach to bioelectronic medicines. Now, the final thing that I would like to talk in terms of is that there is a particular, I mean, many of you must have already seen that at the present moment there are tests that have been done where the brain activity pattern is converted into certain impulses, and that controls the hand motion, or that controls an outside hand and tells it to do what it wants, what we want it to do. So the ability to map the brain potential, brain neuropulses and neural activity, convert it into a significant direct impact actuation system, and then get back. Because suppose you are trying to grab something through your this thing, your only visual clue is through the eyes, so you are guiding it through your brain. It's an open loop system. There's no direct connectivity. The open loop system has its own particular this thing. I mean, neural noise, the noise in the detection system, actuation system problems, and things like that. So when you are trying to talk in terms of this actuator getting actuated by the neural signals, then you understand that the bioelectronics that is implanted in the skull for doing all this particular thing, or that is trying to take from the skull skin and at a micro level depth, then how that potential is being able to convert it into an algorithm and actuating this particular thing. That is the bioelectronics system. Why we are doing it? That tomorrow, if God forbid somebody has lost his limb or some this thing, can we generate artificial nerves system to his hand so that we can make him do the work as actual. So limb prosthetics and other particular things are becoming a possibility and are this thing. The most important part that is happening at the present moment is that are we able to create medical interventions both in terms of diagnostics, disease control, and release of drugs. So the three cases we try to talk in terms of continuous glucose monitoring and insulin pump, dialysis, microdialysis, and creatine movement or creatine measurement. And the third, which we try to talk in terms of how we can have intracellular detection of certain particular molecules or certain particular markers, biomarkers, which have been triggered either through the tumor cell or have been triggered through a similar process as of a tumor cell. And at the same time, are we able to create a parallel domain in terms of system biology, in terms of stem cells, stem cell progression, and how stem cells are bifurcating, and if we implant those stem cells into a particular cell domain, and how it impacts those that cell domain, and how it interacts with them and tries to connect with them, tries to control them, or tries to improve their functioning. That's the other system biological electronic medicines. I think I'll stop here, and I will like to see if any questions are there or any this thing. Hello.
Any question, please?
I don't think I was able to convey much.
Sir, there is a question in the
IoT,
chat box.
yeah. IoT as a nanowire is possible, but the problem with IoT is that there is, at the present moment, no such algorithm available to convert that signal, because IoT is basically an internet-based system. But these nanowires and other particular things are, you can control them by direct connectivity. So you can have not IoT, but IoT-like algorithms connecting and interacting with the nanowires. Nanowire arrays are being used in order to have a certain interaction with outside world. As I said, you have the scalp or EEG, and based on that scalp electronics, you try to control particular things. So you are able to project out the electrical brain activity into an algorithmic outcome data, and that controls an actuator. So that is possible, but not in terms of IoT, because it is not an Internet of Things, because the nanowire or other particular things do not have that connectivity, and it would become risky in order to have that kind of a connectivity to nanowire that is implanted. Is it clear?
Yes, sir. Yes, sir. Thank you.
Yeah, thank you. Any other question? Basu Saab? I think basically because this was more towards biology rather than towards electronics, that's why most of the engineers are not able to go through. But believe me that it is a very, very important area, and I think what we need to do is we need to go into
Yes. I request Professor Hariman Gupta to comment, give his
Yes,
comments.
Professor Tak, I enjoyed your talk. Okay. So
Thank you, sir.
The multidisciplinary talk basically.
Yeah.
And yes, and to understand this, I possibly have to learn the biology again. Possibly not again, means for the first time maybe. Okay. But anyway, this I find that there could be several electrical models could be there, which can be used to imitate some of the activities and try to ascertain the effectiveness of the techniques. That's what my saying.
Definitely. Sir, the simulation modeling of ganglion, simulation modeling of neurons, simulation modeling of their interactivity, and modeling of the interaxial noise and how to suppress that, these particular things are very well reported, and much of the work has been done in this area. The unfortunate thing is that I'm sure there must be a lot of people who are working in this area in India also, especially those who are this thing. But I don't see many people working in electronics and combining with the doctors in order to do that. So that is probably one thing that we need to do. So nice to see you, Professor Gupta.
Yes.
Yes.
In fact, in IIT Delhi, we have a bioscience department.
Yes, sir.
And biotechnology department.
Yes,
both.
sir.
And they are collaborating with AIMS in various kind of situations.
Yes.
Okay. And fairly long back, I also collaborated with the AIMS in image processing area. So therefore, the collaborations are developing. Okay. Only thing that it might be that the stage may be nascent and something like that. I also attended some of the doctoral thesis presentations in which they have the drug effectiveness in cancers. That kind of a thing they were trying to do, but trying to model it basically from the electrical engineering point of view, electronics engineering point of view. That they were trying to model. That basically they were trying to store the energy in, or the energy they were saying that they are putting inside certain capacitor, and capacitor keeps discharging at the rate in which the drug starts flowing. So those kind of models they were trying to develop.
Yeah, that is a very strong area in terms of pain threshold, this thing, etc. How to control pain and how to this thing, drug release models and other particular things. Very correct.
And in fact, if you have heard Professor Karmeshu,
Yeah, very right. Yeah.
Then Professor Karmeshu was supervising one of such theses.
Yeah,
He
he's
was
very
trying to
strong
develop
in
mathematical model.
correct. He's very strong in mathematics, so he could be
Yeah.
doing this particular very well.
Yes.
No, but my problem with at the present moment, sir, as I said, there is a very strong area where people who are working in the electronics, they need to understand the behavior of these micro electrodes, mini electrodes, nano electronics, and how it impacts biology and biological systems.
That's
That
right.
kind of a
Yeah, you are right. You are very correct. Okay, very correct. Okay.
anything else?
Any further question?
I think after me there is
Professor
another.
Krishna, are you talking? Professor Krishna?
No, madam.
Okay, sir.
So then I think we should close this and you can have the second talk.
Okay, thank you, sir. Thank you, Professor Kak, for his valuable time and such a motivating speech.
It's getting midnight here, so that's why I thought I would close down now.
Yes, sir. Obviously.
And thank you, Professor Basu. Thank you, Professor Gupta. Thank you, Deepti. Thank you, Dr. Saritpal. Thank you so much.
Thank you very much.
Pleasure, sir.
Thank you very much,
USA you
sir.
were speaking. Yes. From USA you were speaking. It took a lot of trouble. Thank you very much. Sarit, do you have to say something?
It's okay, sir.
Sarit?
It is
Sarit, say-
such a pleasure to at least meet, even if virtually, so many people. I have probably met you also after 2 years or so more. And Professor Harimon Gupta I probably met after 3 years or more. And Sarit also after 3 years or so. So it's a great pleasure.
Thank you, sir.
Thank you. Bye. Thank you,
Sarit
everybody.
speaks less. Sarit speaks less. Works more.
Yeah.
Yeah, he speaks less. But
I have.
Sarit was your colleague also for
Yes,
some time
of course. Yes.
in the university.
No, he, at various levels. I have seen him at various levels.
Right. Thank you very much, Professor Khan.
Welcome, sir.
Sir, should we start our next session?
Yes.
Yes, we can start.
Our next session will be taken by Dr. Piali Boshak. Ma'am, are you listening?
Yes. Yeah, I'm.
Should we start, ma'am?
Yes, you can start.
Okay. Dr. Piali Boshak is our next speaker. Dr. Boshak is presently working as Associate Professor in School of Bioscience and Engineering, Jadavpur University. She was Junior and Senior Research Fellow of Material Science Center, IIT Kharagpur, West Bengal, India, and completed PhD during this period. The title of the thesis was Polymeric Hydrogel and Polyurethane Matrix for Controlled Drug Release. She worked as biomedical engineer at RTIICS, Kolkata from 2003-4. Lecturer in 2009 and 10 at Biomedical Engineering Department of JIS College of Engineering, Kollani, Nadia, West Bengal, India, and working as assistant professor at School of Bioscience and Engineering, Jadavpur University, Kolkata since 2010. She was selected as UGC Assistant Professor in UGC Faculty Research Program. She is the winner of 2008 Biomaterials Student Travel Award, winner of Young Scientist in Biomaterials Award in RULA International Innovation and Betterment Award Ceremony 2018, and also winner of Innovative Researcher of the Year from RULA Awards in the year 2020. She is the life member of Materials Research Society of India and life member of Indian Science Congress. Dr. Boschak has produced 9 PhD students and presently supervising 5 PhD students. Dr. Boschak will deliver her speech today on applications of electronics in novel drug delivery system. Welcome you, ma'am.
Okay, my slides are visible.
Yes.
Yes, ma'am.
Yes, it is visible.
Okay, thank you. Thank you, Professor Pal, for inviting me to deliver this in this FDP program. And my topic is today a bit related to again to biology, like previous one. So it is the application of not totally biology. It is more concentrated. It was a broad theme previously that a total biological field has been covered. Now it is just will cover some of the small field which is related to the drug delivery system and how this electronics is applied in the drug delivery system and what is the current status up to that. So to go to the introduction, I am just in brief saying that actually what we mean by the term drug, that is written already. You all of we take medicine. It is actually in the language of science called drug, and it is not anything which you consume to do something bad. It is the medicine. Actually, it is the drug. Drug is the substance or product that is used to or intended to be used to modify, explore physiological system and pathological state for the benefit of the recipient. Now, what will benefit the recipient will have that our body maintain a body homeostasis. That is some optimum level is there. Now, if there is something bad, that is, if we cross the optimum level in its higher limit or in the lower limit, then we are becoming ill. Then the body's physiological condition in the sick state. So we need something to get back to that body homeostatic condition. And what we take some medicines, which may be some therapeutic product, which may be from some chemical entity, maybe from a natural product, which take it to get back to our body condition in our optimum range. So it is the medicine. It is the drug. Now, what is the drug delivery system? We take every day, I think most of us take some of the drugs or some of the medicine. We take that a tablet or a capsule. So why there is a question of drug delivery? What is it? It is the process actually. It is the process of administering, and everyone can by themselves can do it. So why need to discuss? Because actually it is the absorption of transport of drug to a particular part of the body. Now what is the particular part? The part which is affected, which needs the treatment. Sometimes it is the whole body, sometimes it is the specific region. So not the taking of the medicine, that portion, the things from the medicine that is actually give you the relief. How it is getting absorbed or how it is giving, after that absorption it will give the action. So it is the absorption and transport. Now this process of the medicine, how it is getting absorbed and how it is getting transport to provide us the medical benefit or medical effect, it is discussed in the topic of drug delivery system. Now, traditionally or conventionally, we know there is a drug delivery system for a long time, because people, whenever get sick, they will try to make themselves to the normal, and in course they will take the medicine in various ways. Now, if there is some scars in your hand, you will take some medicine which is, it may become cream, and well recognized scheme is in at least in Bengal, it is the Borolin, everybody use it. So it is also a drug, and it is also used transdermally, okay, upon the skin. Now, this traditionally this drug delivery system can be subdivided in number of depending on the number of ways. Now, if you consider the route of drug administration, but that is how we are taking. It can be local, it can be systemic. And among these different routes, we mostly use the oral route. It is the more, because it is much convenient, it is easy to use, people can themselves take it. And besides that, there are a number of routes, and this oral route include the systemic route. That is, when you take to the oral route, whole system is getting covered by that therapeutic material or medical medicine. Okay. Now, there may be some local route like topical application, deeper tissue application, arterial supply. That we use this topical applications of the different drugs for a number of times. Like any cream we using. That is the topical application, because we are using locally where it is required. And for systemic application, obviously we can use by ourselves the oral delivery systems, but for sublingual also or buccal delivery system, we can use it by ourselves. But there are other routes like rectal, cutaneous inhalation, nasal, parenteral. Except parenteral, from 1 to 6, all we can use it by ourselves without any specialized training. And among them, most popular is the oral. Now, for parenteral route also, there is some subdivision or subtypes. The subcutaneous, that is just below that skin, and intermuscular, intravenous. One should be very, very much trained to use this intravenous injection, and transdermal injections. These are fall under the parenteral category, and they need some assistance for most of the people. Common people without any training cannot use this route. Among the different route, cutaneous route, that is transdermal, where we are using the skin. That is, we are applying our medicine above the skin, and it will go through the skin to reach our systemic circulation to provide us the systemic effect of the drug. Is another important route. I am highlighting this because there is some application of the electronics on that route. Now, traditionally, what we are using drug, the medicines that can be of various category based on the dosage form also, like tablet, capsule, solution, suspension, emulsion, syrup, elixir, aerosol. We use everything, but sometimes we do not just notice which type of thing we are using. That is for suspension type of medicines. It is always written shake well before use, and for emulsion also. But one should follow it before applying it. Otherwise, we will not get the intended action, which is actually for which we are taking it. Now, when there is some traditional drug delivery system, where is that requirement of the newer delivery system? Why we can use that one? Why to waste time on the newer things? Because there is certain limitations, certain drawbacks, which actually enhance, which actually encourage us to do something better or do something to get rid of these drawbacks. Which are the drawbacks? That they are mainly non-targeted. That is, they mainly systemic. The whole system is actually come under the effect of the drug when you take the medicine. including the site of action where it is needed. So it is non-contagious. Uncontrolled drug release kind of thing. It is not always uncontrolled, but the control what we want, that is not always achieved. It is around that. Now I will explain it further in the coming slide. Besides that, there is something which is called first-part metabolism and effect of GI tag use. Actually, these are the more scientific terms. What is first-pass metabolism? When we take through the oral, that is mostly which the people take. For oral delivery, it is always go through the first-pass metabolism. That is, it is always go through the liver. And liver metabolize a part, and remaining is actually giving us the action. So we have to feed our body a more amount of drug, which is actually required for action. That is why always you will find that injection doses are less compared to the oral doses, because injection is directly going to the system. So there is no first-pass metabolism for the parenteral route. And there are other drawbacks of this first-pass metabolism. There are some very good materials which have some therapeutic activity, but they get totally metabolized by the liver. And so ultimately, through oral dose, we will not get any actions. Besides that, another problem for oral dose is that effect of GI tract juice. GI tract juice means, everybody knows that our GI tract has a pH, it is acidic, it has a pH of around 1.2, and it is highly acidic. So a lot of drugs get deactivated, get decomposed over there, especially protein peptide type of drug. That is why their oral delivery is limited. If you want to get some oral delivery, we have to go for some novel delivery system, some modification. We cannot get it like other tablets through processing. We directly cannot take it, because it will be destroyed by the GI tract juice, and it will cause gastric irritation also. There are some drugs which are acidic in nature, and if you take it orally, you will cause gastric irritation. There will be some burn sensation in our stomach. Due to it will be it is called ion trapping. It will provide that effect of ion trapping in the surrounding gastric mucosa. As a result, there will be some sensation like burning like sensation. So these are the problems already present in our conventional traditional drug delivery system. And there are some ways also to get rid of it. We sometimes coat it, the drug with some coating, so that it will be protected in the GI tract, or we sometimes use its other forms like parenteral delivery form, so that the GI tract and liver could be bypassed. So now, what are the novel things? What are the newer things we're trying to get rid of this thing with combination of electronics? These are the things what I told you. It is written over there. And another important term in this case is that this therapeutic index or therapeutic level. When you take drug, we do not think much of that, all those things, but it is actually happening, and it is actually expressed in drug delivery systems, that the drug should maintain this level. This is the upper, this is the lower. Below that, the level, there is no action of the drug. The intended effect is lost. Above that, it will be toxic. So you have to maintain that level. Now, in conventional, what we take, or we take tablets 3 times a day. First time we are taking tablet, it is going there. Now, before going it down to that lower level, we are taking second dose. It is, so we are maintaining that level, so that we can get action. And for injection, it is at higher level at first, and then it is decreased. Now, what is our desired goal for novel delivery system? What more thing we can want from the newer delivery system? That you want to maintain that level, this level, like that, For how much duration? For which we required. That is, till we are not getting well. Whenever we are getting well, it will be excreted. No drug will be present. So, again another thing, it is the controlled release. That is the right concentration. We are trying to achieve through novel delivery on newer drug delivery system. Trying to achieve mainly 3 R. That is the right concentration, the right place where actually it is needed. Not the whole body should be exposed to the medicine or drug. And right period, that how long it is retained. Till that time drug will be present. So this is the approach for novel delivery system. Now, based on that, we can classify again it rate pre-programmed, activation modulated, site targeted, and feedback regulated. Now, rate pre-programmed, I am not going details. Just remember that when we are taking some tablet or capsules, mostly they are matrix type. That is, matrix means there is some processing ingredient, and within that processing ingredient, drug is evenly dispersed and distributed. And when you take the drug, it will release through that matrix. And what is reservoir? That is, in the reservoir, drug is within the reservoir, and surrounding the drug, there is some covering. Okay, and like some cod liver oil type of capsule. That is, the cod liver oil present within, and surrounding that there is some covering. And whenever the covering is getting dissolved, all drug will be released at once. Okay. So mainly these 2 type of delivery systems are present, and we take everybody has been gone through this type of delivery system at some period of their life. Coming to next. Now is the activation modulated drug delivery system. It is now. It is like same. It can be reservoir type, can be matrix type. But main thing here is that besides this rate controlling surface is present for both reservoir and matrix, here there will be some energy sensor or activation modulator, which will actually control the release. And this can be of different type. This activation modulator can be of different. That can be of osmotic hydrodynamic vapor pressure, or mechanical, like that puff we take. That is the mechanical. Magnetic, it can be iontophoresis. It is the electrical energy we are providing to control the release. There is sonophoresis, that is where we are using the ultrasound. Besides that, chemical activator can be also there, pH and hydrolysis, and biochemical activator will also be there. Now, there is another thing, that is it can be feedback regulated ultimately, because if it is feedback regulated, then only whatever we require, depending on our requirement, that drug release will be controlled. So there will be some biochemical responsive energy sensor, biochemical or responsive, that will actually control the release depending on the system required. System, what is the system? It is the human system requirement, depending on the requirement. Now I am coming to the transdermal drug delivery system. Now it is the delivery of drug through skin. It will be like this band type of thing, and the device is like that, and it is just worn in the hand or like a belt, and through that the delivery of drug will take place with time. There are some benefit also. That is, it is non-invasive. It bypasses the fast metabolism. So the drugs which destroys in the liver or get consumed can be used in this way. And it provides easy control of the drug release. How? I will come in later. It provides sustained and controlled release delivery for long period of time. It avoids the chemical degradation drug in GI tract also. So these are the different benefits where we are using the screen. Now, what are the limitations? The main barrier is that flow of drug to the skin, and in that the main barrier is the stratum corneum, because it is restrict, it is stop the flow of drug to the skin. Now, as you know, this is the different layer of the skin, and among this layer, when you are putting the over here, it should go pass through the all layer to reach the blood vessels, and only after reaching the blood vessel it will provide you the action. So these are the main drawback, that you have to do something so that it's able to cross the layers to reach the blood vessel easily. Now there are different type of transdermal drug delivery techniques. That is, you can skip out the stratum corneum, but it is not a good choice or good solution. Now you can hydrate the stratum corneum. Okay? It will allow you to release a better release of the drug through it. Now, beside that, now how you can enhance more, that is through antiphoresis, to applying some small current, to sonophoresis, to applying some ultrasound, to thermal energy or electrophoresis. There are some chemical approaches to, that is a delipidization of the stratum corneum, but people do not want it to do mostly because they don't want to alter anything unnecessary when we can take the medicine through orally or other ways. Or synthesis of lipophilic analogs, that will also help to go through more easily. And biological approach, use of skin metabolism inhibitor. There is another term actually being for biological approach. There is another term is called, that is the product. Product. What is product? We can just conjugate our therapeutic material with something else and make a product. That is, it is not actually drug now. It will be drug when it will be just displaced from the thing to which it has been conjugated. Until that, it will not provide any action. So it is the product. And there will be some, if you use this product in the skin, that product should be metabolized by the skin in metabolic enzyme. Within the skin, there will be something, some biological element. It will just clip that drug from the conjugate and allow it to grow through the skin and allow it to provide the action. So it is another, you need some more chemical modification, chemical reaction beforehand to provide this product, to get this product. There may be some vesicular approach like microfabricated microneedles, microchips, and these are the recent approach. In micromaterial, what are mostly are made from polymers. In microchip, there is some electronic circuit which will control the release and as per need. Now we are coming to electronic drug delivery system. It is the most recent approach, you can say, among the novel drug delivery system. This is the mass sensitive and accurate delivery. It provides smart, sensitive, and accurate delivery of the drug, more accurate, obviously. And among the electronic drug delivery system, you will find some microchip-based drug delivery system, some electronic capsule-based drug delivery system, and electronic pump is available for long in the drug delivery area. And there is some auto injection. If you are associated with some hospital, you will know that about that syringe pump. It is also some auto injection pump. It is also used for long. And beside that, there is some electronic transdermal patches. Transdermal patches, again, when there is transdermal patch, transdermal delivery system, and we are using electronics, something on that, we can term it as anti-process. Now I am coming to the microchip device. Now, it is drug delivery using the microchip. A microchip, what is the microchip drug delivery? A microchip is an implantable delivery device that stores and releases drugs from small reservoirs. It has a number of small reservoirs. Okay, and this each micro reservoirs it can be associated with same drug or number of drugs, and it has a metal membrane, and it is a capped with a metal membrane, and there is some circuit control by microprocessor or other sources, and that control circuit consists of a timer, demultiplexer, and microprocessor. It also can have a thin film micro battery, which will provide it the power source. And small doses of therapeutic drug or chemicals are stored in the micro reservoir will be released on the signal received from the microprocessors as per the need of the patient. This microprocessor based electric supply could be controlled from outside also. Now, here, which type of drug we can deliver? The drug should be potent and highly soluble, because that microchip should be very small in size, and to enable it, the drug should be very potent. Potent means that is, it will have action which very less amount. That is that volume and amount will be less. That can be only can only be used using this microchip device. Now, it will provide a constant release of drug over a long period of time, and you can control the release as per your need. Now, it has an advantage of better control of dosing. Besides that, it has an added advantage. As we are using this microchip, we can gather information of blood flow characteristics also by using the data collected by it. Now, is this microchip device is a dream or is in reality? These microchip delivery devices are currently being studied in the treatment of some serious diseases such as the breast cancer, multiple sclerosis, osteoporosis, Alzheimer, Parkinson's disease. So it is on the obviously in lab scale. Is it any in the market or any patent has been done? Yes, the first patents on microchip drug delivery has been awarded in 1998, long ago, to a group of scientists from MIT. And after that, a number of patents has been awarded to that same group and other groups also from the other university also. And clinical trials also has been performed. And first human trial was on the human parathyroid hormone fragment was delivered using this microchip based drug delivery device in 8 osteoporotic post-menopausal women subcutaneously, that is just below the skin level. Subcutaneously it has been put, and the delivery has been controlled as per need. Now I am coming to the electronic capsules. It is also almost similar like that microchip, but here we are delivering the one drug. The electronic capsule is an oral electronic drug. So you can use it like orally, like general capsules also. And it can deliver a drug or drugs. And patient, it can monitor also patient. Like only it can also can be used for patient diagnosis and monitoring. The capsule actually contains a microprocessor, a battery, and pH sensor, temperature sensor, and all this transformer, because we wirelessly we actually transfer the data from the capsules to the outside and outside to the capsule. There will be a fluid pump also, a drug reservoir too. It can deliver the drug with specific portions of the GI tract, which is otherwise difficult to achieve. Why it is difficult to achieve? Now, if you go through the nature of the GI tract, you will find the pH of the GI tract is different in different areas, but it is very similar. It is very similar. Why? When from the stomach you reach the small intestine portion, the total intestines, small and large intestine, they have the basic pH. The whole area is basic pH. pH changes, but the total is basic. So by targeting a specific area is based on the pH is very difficult. So it's sometimes difficult to treat some disease associated with that area, like corn disease, like colon cancer. So what happens when target based on the pH, the whole intestine colon get affected with the same drug. But it may not be required for the whole things. It may not be required. It may be required for a specific area of the GI tract. So for that, this electronic capsule is very much useful. So besides that, currently there are some tablet we use for the specifically for this intestine. What we do that we dictate the release initially, because the rate time is fixed. Time is already known that when you take the drug orally, it almost taken 1 hour to reach the stomach, and after that it reaches slowly to the small intestine and large intestine. So what we do, we dictate the release, initial release of the drug, so that it will be released on the intestine again. In that case also, the whole intestine will be exposed. So that is in those cases, this electronic capsule will be very much helpful. And besides that, for some drug like some salicylic acid type of drug, they provide a gastric irritation. So we just coat it with some coating, which is called enteric coating. This enteric coating, what happens? It will just prevent the release of the drug in the gastric area, and when it reaches to the intestine, it will be dissolved and released. But again, the whole intestine will be released, affected. So to get rid of this problem, to specifically target some areas of the intestine, we can use this electronic capsule. Now, there is some disadvantage of this electronic capsules also. That is, it is very costly, high cost. It is there is some manufacturing difficulties. There is biocompatibility issues. There is some danger of device failure. So, but the advantage also is number of advantages. That therapeutic effects are more predictable because it is releases a full dose whenever where it is needed. It can facilitate the control, extended release, and targeted delivery. What all things we actually try to achieve through our novel delivery system, that can be achieved through this electronic capsule. This electronic capsule can communicate data via wireless trans receivers to an external control unit owned by the patient, and it allowed the real-time data recording and wireless remote control of dosing. So, is it these electronics capsules are what is the current status of this electronics capsule? Is it in use or not in use? So I am coming to that. Yes, IntelliCap. This is the world's first oral electronic drug delivery and patient monitoring capsule developed by the Medimetrics. And it is loaded with the therapeutic agent and inserted in a programming station where it takes the account of patient's specific factor. So it is initially fed with the data required. That what is the data required? Because the data will give the electronic capsule the command how to release and when to release, and depending on the patient factors, patient's body weight, patient's age, and where the point of delivery in GI tract, pharmacodynamics of the drugs. when we are using some specific drug, one drug, that drug is pharmacodynamics also fade. If we are using number of drugs pharmacodynamics fade, and how it will be released and keeping some gap or not, or simultaneously, that will also can be controlled beforehand through training it. Once the patient, and now what will the patient do? Once the patient swallows this small single-use capsule, it is designed to detect the stepwise pH, and then by automatic programming of the capsule, dispense the metered dose of the medicine. The capsule follows the elimination process of a regular capsule in the body. So that is one way advantage, one way disadvantage. As it is just following the regular elimination process, so it will be eliminated within 24 hours. So, but it will, within that 24 hours, it will provide accurate delivery and accurate data also. Now, it is also in preclinical studies performed in the Bengal dogs with a reference arm in which the atenolol is a drug is targeted to for the release in the small bowel and colon, indicating that the result were consistent. And it is also has been used, that same Intel cap system has been used as a sampling tool to collect the GI fluid. It is another important issue. And if it could collect the GI fluid, and you can just collect it after it is getting excreted, then we can have that GI fluid and can culture it, can get more data about our system, that is about our health condition of the GI tract. Now, it can be help in effective treatment in different GI disease like cancer, Crohn's disease, intestinal, other intestinal diseases. Now, this programmable technology will be able to individuality. It was initially, if there was some individualized treatment concept, that is treatments required for each patient, but keeping the mass demand, that tablet and capsules for generalized has been developed, and it is easy for individualized. Actually, the cost become very high, but as itself this IntelliCap is a costly thing, so it will provide the individualized treatment to each patient. It is also in the preclinical phase, and it is in the human trial. So, but beside that, what are the electronic drug delivery systems are present beside this microchip and capsules? There is electronic inhaler, there is electronic injection pen. Injection pen is always present, that is it deliver a metered dose, fixed metered dose. When you are using the electronic injection pen, it is like that micropipette like type of thing. In micropipette, we use that required amount, set amount for our laboratory experiment. Here, we are using some needle to put it in our human body, in our body as per requirement. Electronic auto injection is present for long, and electronic wearable infusion pump. These electronic pumps are also used for long, and now they are further modified, and the miniature forms are getting developed. Now, what are the advantage of the different electronic drug delivery system? This advantage of the insulin pump delivery system include the flexibility with the timing of the meal, the real-time measurement of the glucose level, alerts for hypo and hyperglycemia. Now, here is the main important term, that is that we are taking the insulin to treat the hyperglycemia. Sometimes what happened, we don't know it reached the hyperglycemia level, and if there is some hypoglycemic shock, then that is irreversible shock. So anyway, it should not reach the hypoglycemic level. So that is why this alert is very much important. And early notification of the impeding hypo and hypercalcemia. It is an advantage of auto injection in that device are able to hold the liquid and lyophilized formulation, whereas other device may only able to hold the liquid formulations. And this delivery system allows for the fast delivery of emergency therapy and also provide the convenience of self-administration without medical office visit to receive the treatment. And the rate when you are giving the injection by hand, it depends on the person's expertise and at which rate he is pushing the injection. But for auto injection, the rate is always set, so there is it is maintained set. So this is a very much advantage. Besides that, electronic capsule is useful because it can deliver the drug to a specific portion that I already told. And besides that, electronic patches can deliver the multiple drug at once. Obviously, electronic capsules also can deliver a multiple drugs at once as per the requirement. It can also deliver protein peptide as well, because how it can deliver it, because it will limit its delivery in the stomach. It will release its delivery in the area, small intestine, where it will be readily absorbed without any destruction. It also allows for a broader range of drugs to be administered with greater control, which is not possible for the conventional and traditional delivery. For that, we need this electronic help, electronic accuracy. This expands the potential of delivering drug non-invasively and active transport of the drug across the skin. And the electronic transdermal patch is a painless alternative to injection and offers a more immediate onset of action for relief. Now, this is this applicable for every type of disease or every type of drug? No, it is not that like that. It is mostly applicable for insulin delivery system, because insulin you cannot take away orally; it will get destroyed in GI tract, except from few drugs which are just coated or modified so that it will be not be to that protect from the GI tract. For those type of insulin delivery, we mostly found that there is some pain or injection, and instead of this pain injection, we can opt for the transdermal delivery, where we can use this electronics to get better delivery of drug. Why? Because in transdermal delivery, the stratum corneal or skin prevent the passage of the drug through the skin, and to enhance that, we need some small flow of blood. And there is, you will find there is that estimated projected delivery market has been increasing day by day because of this, those advantages told earlier. Now there is some Renesas electronic drug delivery system is already present in the market, and it uses some two microchips, and it is mainly used for the insulin pump or infusion pump to control the meter dose specifically as per requirement. And the electronics part actually, they are using some lithium ion battery MOSFET bridge and DC-DC regulator. And after that, I am again to the iontophoresis, and that is the electronic transdermal drug delivery system. In transdermal, it only we are using skin for delivery of that. Now, iontophoresis means we are using skin, but using some electrical energy. Now, it is a non-invasive method of propelling high concentration of the charged substance. The medical entity we are using or medicine we are using, that should be charged to get better effect of the iontophoresis. That is another thing. Otherwise, you have to just conjugate with something which could be ionized in ionized form, and better effect is observed from the charged substance, and which are the not charged substance, they also get more release or more enhanced uh, passage through the skin. That is also has been observed. Now we are using some small electric charge, approximately 0.5 milliampere per centimeter square. This amount is very much important because everybody knows that there is some limit of flow of current through the skin. So it should not cross that limit. And that has to be, one have to be keep in mind on developing some anti-fluorescence drug delivery system. What we do actually, we do that a drug is placed in the skin, and above which some electrode are placed, and there is another counter electrode is placed. Depending on the charge of the drug ion, actually, which type of electrode will be placed, it is decided. And when you just initiate the flow, it will just enhance the drug flow through the skin, and drug is getting absorbed to the blood vessels. This is the main actually principle of the antiphoresis. So now, beside antiphoresis, how could you improve the or enhance the flow of the drug to the skins? It can be besides, or it can be in conjunction. That is, along with, if you use some thing like that, some other things which will further enhance the flow of drug through the skins, like some gel type of material, some hydration of the skin. It will further enhance the permeation of the skin, and just we will get rid of these barriers, and it will enhance the passage of the drugs to the skin. And so, which type of materials are very much helpful or handy for this antiphoresis? And this is protein peptide type of drug and ionic drugs, which has a very high fast metabolism. They are used for delivery, like antiphoresis delivery. And so here also we use the principle of like charge repel each other. So opposite charges attract each other, and based on that we place this anode and cathode on the based on the type of the drug molecule, which type of drug molecule we are using. A positive ion will be delivered from the positive electrode, and a negative ion will be delivered to the negative electrode. So if the drug which are delivering, we are depending on the nature, we have to place the electrode. So for patient, what will patient do? They have to go through the instruction very carefully, otherwise there will be no delivery of the drug. Now, if you go to the history, is this antiphoresis a new thing? No, it is not at all a new thing. It is introduced in the late back in 1747, and the name antiphoresis has been given afterwards. Previously, it was known as the cataphoresis. It was, but this ions has been delivered by the application of the electricity for long. Now, after that, this term antiphoresis came in the 20th century, and sometimes researchers also term is electrically assisted transdermal delivery. Nowadays, this delivery is getting very much attention of the researcher, and it is exploited for different type of application, like with laser Doppler technology. It is also we are using in combination with the laser Doppler technology as a diagnostic tool also. Now, how this antiphoresis enhances the transdermal delivery? Now it is actually follow mainly three mechanisms. Ionoelectric field interaction provides an additional force that drives the ions through the skin, and the flow of the electric current increases the permeability of the skin. That is, it is sometimes also termed, I have told earlier also in some slide, that is called electroporation. And electroosmosis produces the bulk motion of the solvent that carries the ions or neutral species also. So these neutral species are actually transferred through this bulk motion of the solvent, with the solvent then stream. An electroosmotic flow occurs in a variety of membranes and is in the same direction as the flow of the counter ion. And it may assist or hinder the drug transport, obviously, depending on the nature of the drug. So that is the main thing. Most of the case is assist the drug transport. Now we all have gone through that advantage of, I think, transdermal delivery system previously. Now all this advantage of the transdermal delivery system is present in the iontophoresis, that is, it is painless, and it is the if a patient cannot mostly patient scared of the getting injections. So when you are using this transdermal delivery, there is no you can just restrict the injections. You can reduce the risk of infection due to the non-invasive nature of this type of delivery system. And it had the medicaments is delivered directly to the treatment site, could be delivered directly to the treatment site, and it minimizes the tissue trauma from an injection. Is mostly observed in case of the diabetic patients, and this treatment could be completed in minutes, depending on the requirement, and dosage, obviously. Now the most attractive advantage, or which is not present for transdermal, it is that we can control the release of drug through the control of the flow of current. That is, for transdermal, the whatever drug we are providing, that will grow as per its release rate. We cannot control the release rate, but through iontophoresis, we enhance the rate or also control the rate through controlling the flow of current. Now, what are the factor affects the iontophoretic transport? That is, what system we will devise, will fabricate. What are the main factors we keep in mind during development? That is the molecular size and molecular weight of the drug, which is very much important. Charge and concentration, required concentration for the action. That is the therapeutic index of the drug. That is what level of the drug is required in the blood for the action. Now, what is the drug formula? It is pH, ionic, or covalent present, or it is the viscous. Now, biological parameter, it is inter-subject variability will there. So, personalized delivery or personalized modification of the system or rate could be done. And obviously, where we are intending for. Regional blood flow actually different. That is why this rate of release will be, required rate of release will be different for different part of the body where we are trying to use. If we are using the hand skin always, then the rate will be fixed. But if we want to use the same thing in other places, then the rate will be controlled. Now, what are the electrical parameters important? That is the current strength, current density, pulse current, duration of the applied current, and electrode material. These are the 5 main important parameters for the electrical parameter considered. Now, how this? What are the pathway to cross the skin? If you go through the skin structure, there are different routes. One is the transphalangeal route, and another is transepidermal route. Transphalangeal route is the via sweat gland and hair follicle. Now, what is these are the opening. So, is it always these portions are taken, or it is easy to go through that? Or it is easy to go to the transepidermal, which is the transcellular or intercellular. For the hair follicle and sweat gland, openings is always there, but the point is, through within our system, there is some sweat has already been prepared. There is some follicles, there is some fluid already present, which is coming out of this hair follicle. There is sweat which is coming out of the sweat gland. So there is some negative pressure which actually hinders the flow of the ions through this route. So mainly they go through the intra or intercellular path, that is through the cells, or this intercellular path, or through the transcellular path. Now the laws follows, that is law of diffusion. This is the fixed law of diffusion is followed. Most of the drug delivery system follows the fixed law of diffusion, and for the diffusion of the drug or release of the drug, and Nernst-Planck equation is followed as far as the electric or ionic concentration is present. So the flux due to the electric current, flux due to the passive delivery of the through the skin, flux due to the convective transport due to the electroosmosis are all responsible for this iontophoretic flow of the current or flow of the drugs. Now, pharmacokinetics of the ion transverse, it is the that I told earlier also, but there is something more, that is the transdermal antiphoresis device medication at a constant rate, so that effective plasma concentration remain within the therapeutic window for a extended period of time. That is that we should maintain the therapeutic index of the specific drug we are applying. That should be our main goal for the novel delivery system. Keeping that in mind, we have to set our electric current, or we have to have some measure to just alter the current amount and to alter the release rate. Again, within the tissue, how this movement of the ions taking place. I'm not going into the details of that. Now, what are the ideal characteristics of the drug for antiphoresis? It should not cause any skin irritation. This is another drawback, you can say. The drug nature should be say of like that. It will not show any skin irritation. And it's better if it is low molecular weight. But mostly protein peptide, especially, are not of the low molecular weight. And the low melting point is mostly achieved for the drugs, and area of the diameter of these electrodes should not cover more than 50 centimeters square meter. Otherwise, there is skin burn possibility will be there, and other complication of the skin can arise. These are the requirements actually. Now, what are the drugs used? There are a number of drugs which are been tried for different animal model used for this antiphoresis delivery, and they got a better result also. And here you will find somewhere in some of the studies that there is something called fan diffusion cell has been used. It is nothing but a device where in vitro means outside the body in the lab, we perform the release studies where it is the arrangement. Fan diffusion cell is already has been established device to study the release for the antiphoretic or transdermal drug delivery. Now, device is look like that here. Now, this device produces a continuous direct current and assures unidirectional flow of ions. Now, the features are intensity should be controlled. Maximum 5 milliampere per current you can allow to flow, not above that. So 1 to 5 ampere, their current flow is there, and if you change the current flow, the release rate will be changed. And what is the timer? Timer means how long at a time. And it has been observed that up to 25 minutes, if you just perform this antiphlosis through skin, there is no skin damage, there is no skin problem. Now, here another important issue that when you are using direct current, it is just getting electroporation of the skin is getting taking place, and sometimes the skin getting saturated. So you can use the alternative current also. So there will be a reversal of the skin biology, skin things to provide you better release characteristics. The electrode is used to is silver chloride electrode is the best electrode. But besides silver chloride, you can use for experimental purpose aluminum foil, and for better result platinum electrode also can be used. And these are commercially available electrode, and this type of the electrode used for different assays, different diagnosis purpose. That electrode you can use also here, which may be semi-permeable also in nature, yeah, for antiphoresis drug delivery. So there are different company actually already present in market which provide the electrodes. Now, commercially developed antiphoretic delivery are Lidosite, Forisor, and Itrans, and Forisor 2. So they are commercially available, and they defy the lidocaine, the anesthetic agent, and botulinum molecules, which is used for the treatment of a hyperhidrosis. Hyperhidrosis means that there is a hydration of the hands. So to treat that, this type of antiphoretic delivery system has been used. Besides those system to deliver the fentanyl and antocaines, this type of antiphoretic delivery system has been in use and already available in the market, commercially available. Now there are some type of antiphoresis system also. That is, there is something called reversal antiphoresis. It also find a application in a number of areas. And there is some pulsatile switching antiphoresis where you are using the either DC current in alternating ways, or you can use the AC current also in this pulsating or switching antiphoresis. And antiphoresis and electroporation combination you can also be used. So the design of the instrument actually, so what we do, the power source, then wave generator. Actually, what we do, we can use number of waveforms. That can be AC waveform, that can be triangular, square, and we can find which one will be better. We can use a half wave rectifier, amplifier, filter regulator, then voltage to current converter. There will be some switching circuit also, and the timer circuit also, and that will be connected to the electrode. The instrumentation some look like that, and for the antiphoretic control system, and you can find some ICs over here. And what antiphoretic system has been developed in the labs, in our local labs, has been done with using this IC. You may have better ICs, better options, but with this ICs we have developed some antiphoretic system which have given some this square, rectangular, and AC alternative waveform, which has been fed to our electrode to perform the drug delivery and the process drug delivery. And there are some timer switching circuited also to control the delivery and to just change the different through the different waveforms. And it has been observed and it has been reported also for a number of cases also that through antiphlosis the release rates had been increased to the skin, and there are other problems like beta cells failure could be also be um, for which we actually use this insulin delivery could be overcome without using injection through antiphoresis delivery system. And when you are using the antiphoresis delivery system, the insulin resistance occurrence has been reported very less. And from some research data, it has been observed. You can go through this table, and you will find that scoring what has been given that antiphoresis system is comparatively better to the other systems because this what is the given in strongly negative, that is portal delivery. Portal delivery is only observed for the oral delivery. For all other delivery system, it is not actually observed. So only the for antiphoresis is also not observed. So comparing to the other systems, it is providing better result for the insulin administration. And the dose is calculated using the equation Faraday's using the Faraday's law. And insulin delivery by antiphoresis has been observed that blood glucose level decrease in blood glucose level is earlier compared to the where we are not using antiphoresis, only transdermal delivery is used. But using antiphoresis, the rate of decrease in blood glucose level, that is to get rid of the diabetic condition, is much earlier. And this system has been developed in our lab, and this is the fine division cell we have used to perform the in vitro studies. Here the electrode has been placed, and this is the sampling port. And there are some other applications of the clinical antiphoresis delivery system, like in dentistry, in hypersensitive dentin, in oral cancer in treating, and some local anesthesia. In dermatology, also antiphoresis has been used and successfully. In laryngology, the antiphoresis has been used, and in ophthalmology also antiphoresis has been used. In ophthalmology, the use of the antiphoresis is somewhat restricted because you have to place the electrode in direct contact with the eye. So sometimes a patient become not comfortable with that option. Okay. So there are the merit of antiphoresis. Obviously, we have discussed it earlier also. There are some demerits that, and there are this why I am more interested in demerit, because here we can achieve more. We can have something to do to make it better. So the arrangement to protect from the electric shock. Obviously there is some isolation in all this instrument also. There is isolation circuit is there, and their excessive current density usually result in pain, and this may vary from person to person. So a long database needed to set the range. This is another point where we can work to improve it. And there should not be any burn injury, and for that uh, the electrolyte change and amount of current should be very specifically controlled. Now, any form of the drug in sufficient concentration is necessary for the antiphoretic delivery. It will provide better result actually. Otherwise, non-ionic form it will deliver. It will increase the release rate also. But for ionic, it is more fast. And treatment is somewhat costly because we need an extra electronic circuit or electronic arrangement to assist the delivery. But that is a bit more costly, but that cost with time and with more number of applications could be diminished down. So the conclusion is that it is better than the injection delivery. It can be a better alternative for parenteral delivery, and it bypasses also phosphate metabolism, the GI tract problem, and it could be control the release as per your need. So it can be considered a better option. So these are the files. So thank you for your passion hearing, providing me so much time. So if there is any question, you can ask me now.
Thank you, ma'am. Participants, if you have any question, please.
So it seems that all the participants are like me. Any question? Actually, I also hesitate to ask question when we just attend the FDP. So anyway, thank you. Hopefully, it will help you to do research in some new field like drug delivery system with combination of the electronics. Anyway, if there is no question, so we can leave now. Thank you.
Thank you, ma'am.
Okay.
So the second session has been completed, and we will now end our session here. Our next session will start from second half, that is from 2:30. So please be there, all the participants. Thank you all. you.